So at Rubius, we're creating a new type of cellular therapeutics, and we're using red blood cells as our platform. So I've been invited here today to talk about how we've been able to use Benchling to streamline a lot of our processes. So why do we use red blood cells? Actually, red blood cells have a lot of key properties that are very useful as therapeutics. One is it's a very common cell within our body. We actually create 2.5 million cells per second, so our bodies are actually great bioreactors for this. And each cell, once it's made, can actually last up to 120 days within the body. This means less doses and better patient lifestyles, so they don't have to continually come back for transfusions. Also, red blood cells have a lot of properties that shield them from the immune system. So we could potentially take currently immunogenic proteins and express them in our cells and shield them from the immune system. Another property is that red blood cells are enucleated, so we have very minimal risk of potential transformation when we dose patients with these cells. And finally, the blood transfusion field has actually discovered a lot within typing of blood transfusions and how long we can store these cells, and we can take all those learnings into our processes and say work with O-negative blood and potentially dose 95% of all patients with our therapies. So the way we do this is we take hematopoietic stem cells from a universal donor, and we culture them and differentiate them towards the erythrocytic lineage. Along the way, we engineer them with lentivirus, which encodes for a therapeutic protein, and they start expressing them along the way, and at the end stage, they will enucleate their nucleus along with any sort of engineering DNA, and we will, at the end stage, have a therapeutic red blood cell. The important part is we can go from one donor to potentially hundreds to thousands of doses for other patients. It's a very scalable system. So basically, this platform is very plug and play. We have a vector software shown here, which is whatever therapeutic protein we're interested. And then we have a very standard process of how we culture our cells, and we just plug in whatever new therapeutic we're interested in to our system. And at end stage, we have multiple different prototypes expressed on our cells. So within discovery, what's very important for us is understanding the properties of these cells. So we are very interested in what kind of donor information we're starting with. Donor gender, donor age, any sort of certificate of analysis. And we need to record all that information. We're also interested in the material, the sequences that we're trying to engineer. So our therapeutic DNA sequence, the specific DNA stocks we take this from, any virus we're producing from that. And then along the way, when we're actually differentiating our cells, we take lots of information, such as the expression of our therapeutic protein, any sorts of surface markers of our cells, the expansion counts to make sure our proteins are not actually impacting the health of our cells, and how well our cells can actually enucleate. So within the discovery team, we actually created this new group called Rubius Prime. They are our cell engineering team, and they go all the way from DNA to our engineered cell therapeutic. And our biology groups, including the enzymes, oncology, and immunology teams, basically put in requests into this team saying, we're interested in prototype X, Y, and Z, and they make these cells. And at the end, they pass them back to the biology teams for any sort of biological assay activity validation. So before we met Benchling, we were working on basically a very unstructured informational notebook. So we had a notebook, and we would just put protocols, raw data, analyze data, slide decks, everything. It wasn't very searchable. And we knew we had to create a different system because we were constantly generating all these different prototypes. So along came Benchling, and we found it very useful because we could record all the genetic information that we needed, and that would go into a very nice database of the bioregistry. The biology groups could go into the requesting structure, which would then query the bioregistry saying, I want that material in these cells, and then they can be executed along a workflow which pulls information from the bioregistry and also stores information into the bioregistry. So before Benchling and before the request module, we basically, when requests came in, it was just talking to people or through emails. When we became a little bit more organized, we had Excel logs. But Benchling created this new the request system, which actually helps us determine, what kind of questions are we asking? How many cells do we want? What do we actually want in the cell? What do we want a QC on it? And then we can form a very nice queue so we can see what status our cells are at. This is in progress, this is completed, we haven't even scheduled this yet, and this helps the user understand, oh, when do I need to plan my experiments for upcoming assays? Also, the molecular biology module has been very helpful for us because it's actually accessible to the entire organization. So within discovery, we can design a new construct, and when manufacturing actually has to produce it for our clinical candidate, they can just go into Benchling and find that exact DNA sequence. The module is very powerful. We're able to analyze our sequences, modify them, perform any sorts of alignments on them so we can validate that this is the correct sequence we've been working with. And then we can link these to all the DNA or viruses or subsequent cells we've worked with. And so the really nice thing about this is with the DNA sequence, we can physically link it to batches, shown here. And then when we produce lentivirus from this DNA, we can also link the DNA sequence here and virus batch information here, along with any sort of virus batch information, such as titers. And then finally, it has a very nice physical aliquoting within the bioregistry, so we can see, oh, we have these specific aliquots and tubes that have this virus of this titer that we pulled for this cell engineering. And then finally, the workflow module, is really good for us to build together when we have very discrete parts of our cell engineering system. So we are producing DNA, we're making virus, and then we're engineering the cells. And within the workflow module, we can keep track of that progress the entire time. So basically, when we are making DNA, we are able to pull information from the registry over here, and then any sort of plasmid batches we are able to make from there, we can actually push it back into the registry. And same thing with the cell engineering. We can also register those cells. The really nice thing is along the workflow, when we're profiling the cells, we can also record that type of information. So any surface marker information, any cell counts, that is all recorded and stored into the warehouse. And this creates a really nice, seamless system where we're able to design new prototypes and record them all within Benchling, and it goes into our service team that is able to execute the workflows and bioregistries, and then at the end, any sort of data that we record from it is all linked together within the data warehouse, which is able to be queried in our system. So this has helped us in many ways. It's very convenient for all the scientists and associates in the lab because the user interface is very easy. Actually, uptake has been a lot better than past electronic notebooks. And barcoding has actually kept things a lot easier too, because then scientists don't have to go in and out of the lab to be able to say, I've used this sample. They just scan it, and the sample information is all within there. It has helped a lot with our communication because it's basically one site for all of our key information. It holds all of our sequence information, all of our data generated, and the experimental protocol. So if we have to go in, file IP, and go in and find the protocol, it's all within there. And what's also super nice is all the experiments are linked. So when you use this DNA to create this cell type, that cell type has been put into an assay. We can link all of those together, and we can see where that came from. And this has helped a lot in our flexibility as we have grown in an organization, and because the system is very configurable. So if we have different groups that have different priorities of how they want to organize their workflows, they can also create their own. So it's a very powerful system. The one thing I've heard from most of the scientists is that it's more powerful than they realize all the time. So finally, this has been able to broaden our diverse pipeline, and we'll be moving forward with a first candidate in our rare diseases soon, hopefully next year. So I welcome any questions on how Rubius has been able to implement Benchling. Thank you.
Has your company fully adopted, meaning like everybody in the company uses this, every scientist? That's the first part. Second part is how long did it take, from the time you guys?
So every scientist has not fully adopted this. Within the discovery team and the service teams, they have more adopted it. So within Prime, this is their main electronic notebook. but each team takes some onboarding. So what I find the most useful is when, say, the manager says, I'm only looking at information from Benchling. So I don't want to see word protocols. Send me a Benchling link. And that's a lot easier than, if there's no enforcement, then it'll be hard for adoption, I would say.
Okay. And how did you start the migration process?
Sure. We actually started in a small team first. We started Benchling with Rubius Prime. And then when they sort of adopted it and found it very useful, we expanded it to the other biology teams, and they saw the usefulness of the entire system.
Thank you.
No problem. Oh, dear.
Sorry about that.
Hi, this is Kevin from Finch. I was wondering, Benchling has really great support for their products, but what kind of support structures have you made internally inside of Rubius to help with adoption or with development of the databases?
Sure. So I call Kata our Benchling guru, who basically, she is our source of truth of Benchling internally, and she's the one that interfaces with Benchling in general. And so when people on other teams have questions, they usually will go to Kata and see if that can solve the problem first. Saw a few more hands.
Hi, you mentioned that this was being used to generate the cell lines that are used in the assays. Are you also capturing any of the assay data in this, or is that done in a separate system?
We have assay data within Benchling, but they're in the ELN system, so not in the structured data.
So just related to adoption, what are some of the barriers that you see to complete adoption?
Scientists are creatures of habit, and so if they're used to doing it one way, it's very hard to adopt another. I found that when you need to communicate information fluidly, then Benchling is a lot easier for that, and if that's something you can suggest to different scientists, say, it's actually a lot easier if you use this so I can be able to access the information. That sometimes is helpful.
Hi, you mentioned the data warehouse.
Yes.
So you're using the data warehouse?
We are inputting information into the data warehouse, and we are working on hiring someone in who can write queries. So we've put some things in-house. There are some people on the weekend will program some things to be able to query the warehouse. And it's actually a very powerful system, but probably having a DBA is better. Yeah. Any other questions? Okay. All right. Thank you.